So, dear all, we are just starting with the session of participants, and in this, the first one to present the lecture is Dr. Soumik Goswami. So, he is right now working in Sunbeam College for Women, Varuna, as assistant professor, Department of Zoology, and he is going to present a talk which is related to cisplatin induced toxicity in testis and some of the gonadal physiological functions. So his title is Melatonin Pre-Administration, how it ameliorates the cisplatin-induced impaired steroidogenesis and apoptosis in the testis of Mesochrysatus. And this work was conducted in the lab of Banaras Hindu University under the guidance of Professor Chandana Haldar. And Professor Chandana Haldar, she used to come so much to this department, and we are very thankful to her also that she was always associated with us. Right now, due to some COVID situations, it was difficult to call her, and due to very short notice, we have arranged this seminar. So she couldn't come, but I'm still very thankful to her, and I welcome Dr. Soumik Goswami for his deliberation. And once I would like to tell you that he was a faculty member till last year in our department. So that way also he is very much associated with us. So I wish him all the best. So you can go along, Dr. Soumik.
Thank you, ma'am. First of all, I sincerely acknowledge the Department of Zoology present head, Dr. Tumul Singh, ma'am, for giving me this opportunity to share my personal research work in this national seminar on recent trends in zoological sciences with a theme on nurturing science through. Now, this is how we learn. Now, this is a stage which I was hearing from the morning. This was shared by all stalwarts from the field of biological sciences. All were my ex or former teachers. So it would be an audacity for me to share the same stage. But still, I think I have learned at least something from them so that I can speak in front of some people regarding my research. So this particular study, which is a part of my postdoctoral research work, in which we tried to see cisplatin, which is a chemotherapeutic drug, I'll come to that, mediated hepato and testicular toxicities in the golden hamster as a model animal, with the scientific name Mesochrysatus auritus. Now, the liver part I am not showing over here. It's only the testicular part that we are going to concentrate upon. Now, since we all know what is cancer. It's one of the most feared pathological conditions before COVID, obviously. But still now, it's still an important pathological conditions that we can think of. And the main problem that is associated with cancer is what I would like to focus here is this uncontrolled cell division. As very correctly said by, I would like to quote over here Dr. Murray from the University of Harvard. He said that normal cells, they obey very strict rules, divide only when told, die rather than misbehave. So this is what we know. I'll just skip it a bit because since students already know about cell cycle and its regulation, etc., all these things. So every cell in our body, they cover this particular cycle during their whole life processes, which is perfectly regulated in order to form a new cell, and that new cell has all the chromosomal arrangements, we can say, the protein arrangements, we can say, they're all perfect. So what happens in case of a cancer cell is, so if you see, there are different checkpoints which actually regulate. So what happens in a cancer is this regulation is gone. So when there is no regulation, it keeps on dividing for an infinite amount of time, and thus we get a tumor or something. which is dividing uncontrollably. Now, this shows the global cancer incidences, and mind it, it's 18.1 million new cancer cases. Now, this data is for 2018, so it's a yearly data by WHO, and we can see that Asia has a major share of these cases. So it's definitely an important case for the entire world and for Asia also and India also. Now, if you look at the possible treatments of cancer therapy, you have hormone therapies, early stages, you can have surgery for organs which can be surgically removed, not for every organ. We can have bone marrow transplantations, various therapies, but out of which the chemotherapeutic approach is the most accepted and we can say comparatively cheaper approach towards a treatment of cancer. And in this chemotherapy, this particular drug, cisplatin, it's a containing compound. This made a magical difference in the field of cancer biology. Because 1978 was the year when it was the first FDA approved platinum drug, and it literally revolutionized the field of chemotherapy. So how does it work? If we can just see for a bit that since it contains platinum over here, what these platinum based drugs they do is they primarily bind with the guanine residues. The guanine is a nitrogenous base, we all know, in the DNA. So obviously the cancer cells are like normal cells, they also have DNA. So it binds to the guanine residues and they create some adducts in the DNA molecule, which ultimately interferes with the normal cell cycle and results in the killing of the cells. So the neoplastic effect of the platinum containing compounds are towards here. Fine. This was a revolution in the field of chemotherapy. But everything comes at a cost. So these are the costs that cisplatin and subsequent other drugs containing platinum like carboplatin, they had these severe side effects, out of which the nephrotoxicity and the hepatotoxicity, these two were the predominantly much of concern, because ultimately in the long run, if a patient happens to recover from cancer and develops a kidney associated problem, then that is of no use. So this made the use of this drug a bit compromised, we can say. So what we try to see, so this was just a picture to make you aware of what cisplatin is. So what we try to do, we try to see the effect of cisplatin at the level of the gonads, because that is also a metabolically important tissue, and sterility and infertility, these are also an area of major concern. So we divided our studies into 4 groups, you can see over here. One group served as the control animal, which is for every experiment you should have a control. So this is a vehicle treated control. So for students, I would like to say that vehicle treated control is the animal which receives everything except the drug. Means the animal needs to be handled, the animals needs to be injected with the same solution in which cisplatin would be prepared. Okay, so it's an exact copy of the treatment group, except for the fact that the drug is missing. This next group is the drug induced, that is the cisplatin induced animal. Now we come to the next part where we try to use the hormone melatonin as a alleviating agent, or a possible agent which can be used as a combinatorial therapy with cisplatin, by which we can neutralize the harmful effects. So we had a simple group which was injected only with melatonin, and a 4th group which was acting as a pre-treatment group. That means it was injected with melatonin starting 1 day prior to the cisplatin injections, and then both the drugs they were given at the same animal at the same time. So the whole treatment carried for 4 days, and after 4 days we sacrificed and dissected the animals after 48 hours of the last injection. Now how did we determine this thing? Obviously, we had done a dose dependent study. I cannot show every data over here. And obviously, we had also seen the effect, means we had sacrificed at 24, 48, 72, 3 time periods. And this 48 was the one which showed the predominant effect. That's why we selected the 48 hours group. And accordingly, we had selected the testis for our general histological studies, Western blottings, biochemical estimations of antioxidant enzymes, etc. And we had gone for a serum isolation to do a colorimetric ELISA for testosterone. Moving to the data part. So this is the first thing which we noticed, that this is the body weight in the 4 groups, and we had seen a significant reduction in the body weight in the animals after a treatment of cisplatin. So this green one over here, this shows the before the CP treatment. The red one is after the CP treatment. Now you can see very well in this group, the CP treatment had a significant level of decrease in the body weight. Now, when we dissected the animal, this photo can give you a clearer picture. This is a vehicle treated control animal. So you can see the normal morphology or the anatomical positioning of the testis, and this is the testis of the cisplatin treated animal. So there's a marked regression, which was obviously evident in the paired testis weight that we had also calculated from the different groups. Moving on, we tried to see what was the preliminary effect due to. So we had checked some data on the antioxidant status, as we wanted to see whether free radicals was the thing which is responsible for exerting these deleterious effects. So first, this was a T-bars. T-bars is a product of lipid peroxidation. This happens due to free radical mediated toxicities. So this was raised in the CP group, and promisingly it was recovered, at least we can say partially, in the melatonin pre-treated group when it was provided before cisplatin. The same thing we wanted to check for 2 antioxidant enzymes. Here we have the data for superoxide dismutase, and here we have the data for an indirect assay of catalase, wherein we check, it's an endpoint assay, so we check the millimolar concentration of hydrogen peroxide left. That is, it's a reciprocal type of a relation. If the hydrogen peroxide is more in the sample, that means the catalase activity is less, and vice versa. So you can see over here the superoxide dismutase expression, not the expression, sorry, the activity, it goes down. And similarly, the catalase activity also goes down, which shows a high concentration of millimolar concentration of hydrogen peroxide. So this shows that definitely the primary mode could have been a free radical mediated cellular toxicity, that we can say. The ELISA for testosterone also followed a same pattern. In the CP group, in the blood, the circulatory testosterone level was severely down, which was again recovered in case of the MEL plus CP injections. Obviously, we went for the histological analysis. The first, we have 2 pictures side by side in different magnification. So this is a 10x magnified picture, 10 times magnified picture, and this is a under the 40x objective. Okay, so accordingly we can see these are the normal vehicle treated controls. So in these 2 slides you can see perfectly aligned. The GE stands for the germinal epithelium of the testis. You can see spermatozoa in the lumen of the testis. All the germ cells are sequentially arranged as it should be. Come to the next, C and D. This is for the CP treatment. Now very clearly we can see that the lumen is now entirely devoid of any spermatozoa. First. Next, we can see an increased level of vacuolization. This V stands for vacuolization, which is present in the germinal epithelium and the interstitium of the seminiferous tubules also. So this vacuolization is a marker for, we can say, testicular damage and germ cell death. So here is a condition wherein the seminiferous tubules are not in a normal state. Again, this is the melatonin group. It's more or less similar to the control, where you have again normal levels. And this is the recovery part, that is the pre-treatment part, where we can see, although it is not exactly similar to the control, but it is still much better than the cisplatin group. So there is partial recovery of the spermatogenesis, we can say. Now this might be due to the elevation in the level of testosterone, because that is acting as a direct factor. So that can be one of the reason, or it might be due to the alleviation of the free radical load. So both can be a possibility. So we went ahead, we went for some protein expression analysis. This is the AR, this is the androgen receptor, which is primarily expressed in the interstitial cells of Leydig. This also went down when we checked for the cisplatin treated group. So this shows that not only testosterone but its associated receptor also showed a decreased pattern of expression. So even if the hormone is present and if the receptor is down regulated, still there is no functionality. Further, we went for the steroidogenic parameters, like P450-SCC is a basic enzyme which converts cholesterol to pregnenolone. So we check for the expression of P450. STAR is a steroid acute regulatory protein. This is useful for the transport and the trafficking of cholesterol. And PCNA is a general marker for cellular multiplication or cell division. Okay, so these PCNA levels went down, which shows that the proliferation of germ cells is affected somehow. When we look at the steroidogenic enzyme P450 and in the secondary axis the STAR, it followed a similar pattern. And then we took the cell protective factor, that is BCL2, and a transcription factor NRF2, which is associated with this redox homeostasis of cells. And the NRF2 actually, this is for the students, the NRF2 actually plays a protective part, because this is reported to elevate the activity of the antioxidant enzymes in the cells. So when the expression of NRF2 goes down, so this can be very well correlated with the enzymatic data that I had shown earlier. Okay, so all this in a nutshell, if we can try to see it in a picture, what it shows that this cisplatin, when injected, it induced oxidative stress, led to the downregulation of NRF2, increase in the reactive oxygen species. Obviously, downregulation of the steroidogenic enzymes, receptor expressions for the testosterone receptor, ultimately leading to germ cell death and testicular toxicity, which was evident in the paired testis weight and the circulatory testosterone levels. Now, this a whole picture can be neutralized to an extent if we use melatonin as a supplement. I am using the word supplement. It's not a drug. It should be used as a pre-treatment. Okay, please don't get me wrong. The post-treatments of melatonin in any cases has been reported to be much less effective. So it has to be present before actually the problem comes into picture. So this pre-treatment of melatonin, since it's a beautiful scavenger of free radicals, and it also can activate the expression of genes of these antioxidant enzymes via an NRF2 mediated pathway, we can say that melatonin, if given as a combinatorial therapy with cisplatin, it can really make use of cisplatin as a drug and neutralize the toxicity of cisplatin. So this was what I wanted to share with a very short time. So this part is very important, the acknowledgement part. This I acknowledge Dr. D.S. Kotari postdoctoral fellowship scheme under UGC for providing me the fellowship. Alexander von Humboldt Foundation, Bonn, Germany, for the instrument gift to Professor Chandana Halda ma'am. And I would like to also thank two universities. Obviously, one is BHU, the other is Vishwabharati Shanti Niketan, because I started my PDF work over here. Then due to the superannuation of my mentor, I had to change my workplace back to Institute of Medical Sciences, BHU, for the last year of the PDF. So I thank all of them. They provided me the platform. And since I would like to invite any questions, if any. Since it's online, I don't think questions will come. So we can straight away move to the thank you. ठीक हिस्ट्री एनवायरमेंट